Continuation of the previous video. Molecular dating sources of error. Both x and y values only estimate. Substitution model could be incorrect. Tree could be incorrect. Errors in orthology, assignment. Poisson variation is large. Pairwise divergence correlated. Systematics 53.4. Inflates correlation between divergence and time. Sometimes calibrations correlated. If using derived calibration points, error in inferring slope, confidence interval for predictions much larger than confidence interval for slope. Rate heterogeneity. Rate of molecular evolution can differ between nucleotide positions, genes, genomic regions, genomes, nuclear versus organelle species over time, if not considered, introduces bias into time estimates. Rate heterogeneity among lineages. Cause and reason. Repair equipment. RNA virus have error-prone polymerase. Metabolic rate. More free radicals. Generation time. Copies DNA more frequently. Population size affects mutation fixation rate. Local clocks. Closely related species often share similar properties, likely to have similar rates. For example, murid rodents on average 2 to 6 times faster than apes and humans. Mouse and rat rates are nearly equal. Rate changes within a lineage. Cause population size changes. Reason genetic drift. More likely to fix neutral alleles in small populations. Strength of selection changes over time. New role or environment. Gene duplication. Change in another gene. Strength of selection changes over time. New role or environment. Gene duplication. Change in another gene. Then working around the rate heterogeneity. Identify lineages that deviate and remove them. Quantify degree of rate variation to put limits on possible divergence dates. Require several calibration dates, not always available. Gives very conservative estimates of molecular dates. Explicitly model rate variation. Search for genes with uniform rate across taxa. Many clock tests. Relative rate tests compare rates of system nodes using an outgroup. Then Tajima test, number of sites in which characters shared by outgroup and only one of the two in-groups should be equal for both in-groups. Branch length test, deviation of distance from root to leaf compared to average distance. Then likelihood ratio test, identifies deviance from clock but not the deviant sequences. Likelihood ratio test. Estimate a phylogeny under molecular clock and without it. Root to tip distances must be equal. Difference in likelihood 2 chi-square and minus degrees of freedom. Asymptotically when models are tested, when nested parameters are in set to boundary. Then relative rates test. Tests whether a distance between two taxa and an outgroup are equal or average rate of two clades versus an outgroup. Need to compute expected variance. Many triples to consider and are not independent. Lacks power, ESP, short sequences, low rates of sequence change. Given length and number of variable sites in typical sequences used for dating says unlikely to detect moderate variation between lineages, likely to result in substantial error in data estimates. Modeling data variation, rate variation, relaxing the molecular clock. Learn rates and times, not just branch lengths. Assume root to tip times equal. Allow different rates on different branches. Rates of descendants correlate with that of common ancestor. Then restricts choice of rates but not too much flexibility to choose rates as well. Relaxing the molecular clock. Likelihood analysis. Assign each branch a rate parameter. Explosion of parameters not realistic. User can partition branches based on domain knowledge. Rates of partitions are independent, non-parametric methods, smooth rates along tree. Then Bayesian approach, stochastic model of evolutionary change, prior distribution of rates, Bayes' theorem, MCMC. Then parsimonious approaches, Sanderson 1997-2002, infer branch lengths via parsimony, fit divergence times to minimize difference between rates in successive branches. Cutler in 2000, infer branch lengths via parsimony, rates drawn from a normal distribution, negative rates set to 0. Next, parsimonious approaches. Then Cutler 2009. Infer branch lengths via parsimony, rates drawn from normal distribution, negative rates set to 0. Then Bayesian approaches, learn rates, times and substitution parameters simultaneously. Device model of relationship between rates, Thron, Kishnew et al. Assigns new rates to descendant lineages from a log normal distribution with mean equal to ancestral rate and variance increasing with branch length. Then Wilson-Beck et al. Poisson process generates random rate changes along tree. New rate is current rate, gamma distributed random variable. Comparison of likelihood and Bayesian approaches for estimating divergence times, Young and Yoda 2003, analyzed 2 mitochondrial genes. Each codon position treated separately, tested different model assumptions, used 7 calibration points. Neither model reliable when using only one codon position, using a single model for all positions. Results similar for both methods. Using the most complex model, use separate parameters for each codon position. Could use codon model. Sources of error or variation. Lack of rate consistency due to lineage, population size or selection effects, wrong assumptions in evolutionary model, errors in orthology assignment, incorrect tree, stochastic variability, imprecision of calibration points, imprecision of regression, human sloppiness in analysis, self-fulfilling prophecies. Reading the entrails of chickens, Gore and Martin 2004. Single calibration point, error bars removed from calibration points, standard error bars instead of 95% confidence interval, secondary or tertiary calibration points treated as reliable and precise based on incorrect initial estimates, variance increased with distance from original estimate, few proteins used. Then multiple gene loci. Trying to estimate time of divergence from one protein is like trying to estimate the average height of humans by measuring one human. 1. Use multiple genes and multiple calibration points. Even so, be very molecular times. Point estimates are absurd. Sample errors often based only on the difference between estimates in the same study. Even estimates with confidence intervals unlikely to really capture all sources of variance. MC Leisant, Hockamp, Wolf 2002, dating human gene duplications. Trees generated. Alpha parameter for gamma distribution learned, thrown out of variance mean why would this happen? May be problematic to apply this model for gene family evolution because of possible functional divergence among paralox genes. Then trees built from gamma corrected distances, 2 cluster test of rate consistency. Then balance Hockamp Wolf dating Arabidopsis duplications, create nucleotide alignments, estimate level of synonymous substitutions, Ks values greater than 10 ignored, why used different methods than for human, how reliable is ranking of Ks values, how much variance expected. K greater than 10 unreliable. Yang abstract calculates effect of evolutionary rate on accuracy of phylogenetic reconstruction. Anisimova calculates accuracy and power of LRT in detecting adaptive molecular evolution. Neither seems to have any cutoff regarding DS greater than 10. Future improvements: calculate accurate confidence intervals taking into account multiple sources of variance. Novel models that account for variation in rates between taxa. Build explicit models that predict rates based on an understanding of the underlying processes that generate differences in substitution rates. General references: